where we left off last time was this idea that genome engineering can enable unusual chemistry. So you can see, I think last time I concluded sort of describing these 21 different chemical functional groups that are involved in pretty heavily in organic chemistry, and how the different functional groups boxed in gold are biochemically restricted in common microbes. And so what I'd like to do for most of today, so today will be fully on genome engineering applications. I think based on the way that time has been going and based on my experience and knowledge in this particular area, we will probably spend the entire class period just going through the remainder of these slides, especially since we're just on slide 3 here. And so what I'd like to do today is tell you a story about how genome engineering enabled microbial production of aldehydes. This is work largely that I did personally as a grad student, I guess about 6 or 7 years ago now. And it was a different kind of metabolic engineering. What I hope you take away from this story is that native metabolic processes can interfere with what you might be trying to achieve. And that was not necessarily a guiding principle associated with the development of metabolic flux analysis, etc. I mean, there was the perspective that you could have fluxes redirected through other known pathways. What I'm going to be talking to you more about is that you could have certain categories of enzymes, or rather you could have certain categories of chemicals that you might be trying to make, where you have native enzymes that aren't even known or well documented, potentially have a cross reactivity with a new kind of molecule that you might introduce into the cell, particularly if you want to make something that's non-native. And that kind of competition or byproduct formation can occur in ways that aren't well documented. So when we think about trying to push biosynthesis to the next frontier, or to its ultimate frontier, where it can become as powerful as organic synthesis, and potentially, if not access all of these kind of chemical functional groups, which might not make sense because some organic building blocks and some functionalities just won't be compatible with an aqueous environment. But if you think about just the broader array of chemistry that we would want in our materials and our other kinds of products, pharmaceuticals, for example, it might not just be a matter of finding the right enzyme that can make those. That's very important too. And that's certainly a frontier area within metabolic engineering. But it might also be identifying or creating the right host strain that can overproduce them and keep them around and tolerate them, etc. So with that, I want to introduce aldehydes as a case study. And so the thing to know about aldehydes is that they're a class of chemicals with many industrial uses. There are big differences in the properties of an aldehyde based on its full chemical structure. For example, if we talk about the shortest aldehyde, formaldehyde, which you might remember from any experiences that you had doing dissections in elementary school, or maybe in modern times they've removed formaldehyde as a preservative. Well, it's something that's curious because of both its toxicity, at least after long-term exposure by inhalation, but also its presence in not just industrial uses, but actually as an intermediate in cellular metabolism, even in humans to a small degree. So it's obviously tolerated under some conditions. What I'm going to be talking about here is not really formaldehyde, but it's a useful example to think about because it might be one of the aldehydes that nature, considered in its evolution of enzymes that would try to get rid of aldehydes quickly because of reactivity, toxicity, etc. aldehydes aren't all equal, but when we talk about larger aldehydes, particularly those that are aromatic, like shown here, benzaldehyde or vanillin, as these images suggest, they can have different properties, particularly useful in flavors, fragrances, cosmetics. And these are industries where synthetic biology and metabolic engineering is having a big impact. These tend to be products that are not sold as commodities. They could be, depending on the exact pricing and mixture and application, a specialty chemical category. But if you imagine something like a high-end perfume, the intuition would tell us that that might be something that's quite expensive. And so that could be an opportunity, especially as you want sort of nature-derived, even if using biotechnology type of ingredients, maybe you'd have a preference for those over petroleum. Maybe you'd really try to mimic some naturally occurring chemistries. At the same time, when you think of an actual fragrance or perfume, a very small percentage of it might be your pure product of interest. So these tend to be smaller markets. But regardless, especially in food additives, you've got benzaldehyde and vanillin imparting respectively almond or actually also cherry flavor, and vanillin being, as we've discussed briefly before, the key constituent of vanilla flavoring. And these two compounds are actually two of your most common flavor additives in terms of total market size annually, globally. And so they're of keen interest. And looking beyond just the aromatic aldehydes, if you look at some of the larger or longer carbon chain aliphatic aldehydes, once you get into this range of 5 or 6 carbons all the way to, what could be 12, 14 carbons, you start entering this, well, I smile because I've worked with these compounds in the lab, and it's just really interesting the first time, especially, that I was opening up these powders or solutions and taking in ways that were safe, a whiff of sorts, and being like, oh, this smells like freshly cut green grass or apple. And then you add a couple more carbons and it's more like orange, and then starts to go into lavender. And sometimes these are some of the key molecules that you get, at least in the case of freshly cut green grass. You will actually get some short chain aldehydes that form and that are characteristic of that scent. Interestingly, some of these also can just like that freshly cut grass example might suggest, some of these scents might serve a dual purpose as also like an insect pheromone or some other kind of hormone. In any case, what this map that should be relatively familiar to you by now in this course suggests is that we know how to get to these kind of molecules. We know, for example, that if we want aromatic aldehydes, in general, we're going to go through aromatic amino acid biosynthesis pathways and then make some kind of diversions. Now, you may have already noticed this trend, and maybe I've talked about it earlier, that you'll see a lot of carboxylic acids in metabolism, but not so many aldehydes directly. If you were to envision having an enzyme or different enzymes that could take carboxylic acids and convert them, perform a reduction on the acid group to an aldehyde, then you would get these compounds. And so similarly, with fatty or aliphatic aldehydes, the pathways to make fatty acids are well known. Many of them are natural because the cell has often the C18 or C16 fatty acids that then are esterified and form part of the phospholipid bilayer. There's also been a ton of work in metabolic engineering on achieving different chain lengths, since these fatty acids are assembled 2 by 2 in terms of carbon chain length. So you go from C2 or C4 kind of starting units, and you get eventually to C18, but along the way you've gotten to C8, C10, C12, and you could potentially engineer a way to stop at those lengths or divert some flux. And so people have done that also for fatty alcohols. And in general, in nature, the way that aldehydes frequently appear is as an intermediate to an alcohol. So the biosynthesis part of this equation is not necessarily the problem. As we saw it when I was getting involved really for the first time in this field back in 2010, the challenge was to retain the aldehyde after it's formed. Another challenge that I'm not going to spend too much time talking about that is very dependent on what the rest of the chemical structure is, just like with that formaldehyde example, could be the toxicity of the aldehyde. So aldehyde groups, that functional group is reactive sort of by nature. This kind of terminal carbonyl in general due to its electrophilicity or nucleophilicity makes it more reactive, but it's not necessarily when on certain side chains actually that reactive, or at least not necessarily toxic with endogenous cellular processes. It depends.